Yeah, good evening to all the participants. So today we'll be discussing on mini projects. So let me cover few concepts what is uncovered earlier. But before going to today's session on mini project discussion, can we recap the last session topics? What exactly did we discuss in the last session? And I also mentioned that if you want to do if you are doing some part of a project and if you have any doubts in your project related to bioinformatics, okay, which is pertaining to our syllabus, if you have any doubts, if you want to share the screen and try to clarify, you can clarify it on today or tomorrow. Or else, I can teach you some kind of project skills, okay, from now onwards. So before going to the project discussion, we can also recap the last session contents. Yeah. What are the last session topics we have discussed? So interact with me. So hereafter, the today and tomorrow or the interacting sessions. Remember that if you are not interacting, then I don't think you will benefit much from me. Proteomics, good. We just discussed about proteomics, types of proteomics. What is exactly proteomics? Study about the proteome. If what is proteome? The entire collection of proteins in a cell, or you can also tell like a cluster of proteins grouped in domain. So generally for proteomes, how you get your source? One is through the mass spectrometry experiments. Okay, if it is a peptide, you can also use peptide mass fingerprinting. So these are the techniques, wet lab techniques, which creates the source for proteome. Okay, there is no defined shape for proteome. Very good. So we have discussed about proteomics uh, on yesterday. And what are the other topics? One is proteomics. What are the other? Yeah, only one is interacting, that too with a single word. How about the others? Try to explore because I think after these two sessions, today and tomorrow, you also have an MCQ on the bioinformatics, isn't it? So one should know if only if interact right or wrong, we can correct it now itself. What else did we discuss? Did we discuss something about genomics? Remember the types of genomics? If I ask you what is pan genomics, can someone tell me what is pan genomics? How many genomes are human or initially taken? How many? A question to you is how many genomes, that is human genomes, are taken in consideration to construct first human pan genome? Construct first human pan genome. Anyone know the answer? Just a one word answer. Just because you're not giving more than a one word answer, I'm just given a lengthy question to you. So genomics we discussed a lot. One such genomics is pangenomics. One MCQ I have already leaked. You try this. What is the answer? Give me some random answer, some number, whatever you know. Inky Pinky Ponky. Yes, very good. Even the Inky Pinky Ponky is working right with everyone. So many Pooja Venkatesh, excellent. Okay, the answer is right, perfect. So likewise, okay. So also remember the numbers and if I ask you what is the size of the human genome, immediately you have to come with 3.1 into 10 power 9 base pairs. Okay, so that's what I showed you even on yesterday, isn't it? I just opened the human NCBI genome and even UCSC genome. We found that kind of a size. So whatever is there in the database, we can tell that. Okay, 3.1 into 10 power 9, and how many percentage of coding genes like that. Okay, okay. If you want to discuss anything related to projects, you can post that in the form of a comment. Meanwhile, I take you to the presentation on mini projects. No, no, no. What happened? Something has happened. Okay. I'm taking to the mini project. Yeah, so some of the sponsored projects and research consultancies happened for past 3 years. So these are like in a financial grant I secured from ASD UT. This project is a plant compound, a phytochemical drug. I'm going to target a disease called hepatocellular carcinoma. It's a primary liver cancer. Okay, so for this, I'm utilizing the nanotechnology and computational experiments. Okay, in silico studies. So for this, recently I got like in 2024 to 25 financial grant I secured 5 lakhs, and this is also a currently running project 2021 to 24. So this is a project from a DST SERP. In the subsequent slides, I show you the title. And of course, this is also in silico studies project, a computational study project. And I'm also guiding projects to the other engineering colleges. Okay, and also science and pharmacy colleges. One such project I guided from Sri Krishna Arts and Science. Okay, we guide projects from other institutions in the form of consultancies. We take in the form of a consultancy project and I guide accordingly. Okay. So two MSc students completed a project in informatics, and one was PhD student. The entire PhD topic, whatever she registered in Tech, she changed and she has doing under my guidance at present. Okay. So under a consultancy, so she is almost going to complete her project. She is communicating a publication, and also a few more grants, like even the students those who are doing project under me, they'll be getting a research fellowship. Okay, so around 6.72 lakhs. And even from TNSST, we for the UG projects, I think this is the maximum grant they're funding for the UG projects. Okay, the recent grant. And the Yukti Innovation is one such a competition. Okay, anyone can participate, even UG student, PG student, and even the faculties can participate. And this is the topic I already told. So, out of 1 lakh 5,000 innovations, submissions, only 135 got grants. In the this is I have cleared the phase 4 event. In the phase 3 pitching event, nearly 200 smart India hackathon winners get inside. Okay, in the wild card entry, and they've given the competition. But out of the 200 Smart India Hackathon winners and the 180 something, 180 plus phase 3 winners came into has gone to the phase 4 competition. Okay, I mean the and then only 135 out of 380 people got this grants. Okay, the grant will be from 1 lakh to 10,000. Anyone can try. UG students can try. The purpose of providing this grant is even to enable a startup. So a student can, while studying UG, can be BSc or BTech. Generally, this is from AST. It's better applicable for the engineering institutions, BTech degree and MEM Tech. These students can also apply for this project. And if they get a funding, so after the doing the utilizing money from the funding, they can also apply for the startups. Okay, the minimum 1 lakh and maximum 10 lakhs. Already got a grant, BST grant, so they have reduced grant to 50%, maximum of 5 lakhs. So these are the title of the projects. Okay, so both projects are a slight difference is there. Here I'm using a mitochondrial targeting drug delivery in addition to the in silico studies in BST project. Here I'm using a kind of nanotechnology in addition to the mitochondrial targeting. So presently, I'm working on these two funded projects. And student project competitions under by mentorship. In 2001, there is one Smart India Protein Innovation Challenge. Okay, so my PhD scholar, first PhD scholar, she has cracked this kind of protein hackathon competition. Of course, she has cleared the phase 4 event. Only 20 participants has cracked in India. So she is the top one out of all the 40 winners. I mean, out of all the 20 winners. Okay. This challenge is the India Smart Protein Innovation Challenge. And also in 2000, the ASD, CSCR, the other MOEs, Innovation Cell, Principal Scientific Advisor of the Government of India. Okay, so all these organizations, okay, government organizations um, uh, uh, together initiated a competition called Drug Discovery Hackathon. The purpose of Drug Discovery Hackathon competition is uh, to provide solution for treating SARS-CoV-2 infection. Okay, nearly 30 problem statements are established. Why I'm providing you all this is like even if you're studying or even if a faculty has to utilize this bioinformatics course, and if he or she has to secure grants for the survival, okay, or so I'm giving you all these kind of a tips, okay. So this is it, it happened at that time, okay. Everyone faced a crisis during the pandemic situation, so the government of India came forward with more funding, almost more than 4.5 crores they had for distributing prizes to this competition. So luckily, from Department of Biotechnology SVCE, 12 participants has secured the grant, okay, has secured the internship grant. You know, I've just guided only the six students who got 3.6 lakhs. The other six students is from a different mentor in the same department. So that colleague also, but we didn't receive even a single rupee. The 3.6 lakhs are secured by the students. I just mentored them. Okay, so each and every day I mentored 3 hours for consecutively 3 months. These students are from second year. They don't know even anything about cell biology, bioinformatics, immunology, pharmacology, drug discovery, bioinformatics. They don't know anything. I just started from scrap, okay, and I taken these people to the competition, and luckily these people, each student got nearly 60,000. We may get these kind of a competitions in future, but one should learn all these computational techniques to utilize in the project. Okay, nearly 12,000 plus teams have participated, and only 41 participants has got this kind of a grant. So six students are from my mentorship. 41, I mean, in India. And what are the student funded projects in bioinformatics? This is the title. I told you in Drug Discovery Hackathon, my team of students has cracked the event recently. That is actually where the identifying covalent spike protein inhibitors to control the infection of SARS-CoV-2 from small molecule databases. Okay, so we discovered drugs. Okay, I used techniques like visual screening, molecular docking, MD simulation. Of course, before that, I also gone for ADMET characteristics prediction. So these only four techniques I have used. But even though they have provided me commercial softwares like Discovery Studio, Scroid Engine, I didn't utilize any softwares. We just used only the freewares alone in home. Okay, when we are the pandemic, we are just worked in home, isn't it? So they utilized, and we got this kind of a grant. And even in 2018 to 19, we got a grant for Nipah virus vaccine design, which we worked on reverse vaccinology approach. More about this we'll be seeing in the subsequent slides. Okay, this is uncovered in bioinformatics because this comes under immunoinformatics. That is how to design a vaccine for a virus. Here I have taken an example like Nipah virus. Whatever informatics projects I have completed, uh, 3 years before from Pasteur Institute, Konur. Okay, a PhD scholar has taken this bioinformatics approach, and she has started proceeding working on in vitro and I think even in vivo models. So whatever computational studies we completed, they are taking this as a base, and they are completing the other wet lab experiment. So I think it's going to get wasted. Okay, so whatever we give, they can utilize it in future. Okay, the others can utilize it. So we are giving some kind of inputs to the research society. Consultancy projects to other engineering colleges, not only engineering colleges, arts and science colleges. So this is a project I guided. I'm presently guiding to student, but of course, she is working in the form of a consultancy. Okay. The project is basically a reverse vaccinology approach. Using reverse vaccinology approach, we have to come with a vaccine for hepatitis C. Okay. And similarly, but this project is already completed. That is, this project is, I guided Sri Krishna Arts and Science to two different MSc students. Uh, that is getting a drug, okay, based drug for two important genes linked with pancreatic cancer. So we have gone various computational analysis, like how I mentioned initially, gone for ATMET, virtual screening, molecular docking, MD simulation, etc. Okay. So these are the title of many projects I'm guiding at present. Okay, so these are six major titles. First, genome annotation, function analysis, protein structure. So this is with respect to computational studies. But myself, my PhD scholars are working on in vitro and in vivo in addition to the computational results. We can merge this computational, we can tell this as an amalgamating subject. Whatever computational studies we do, we can do before the in vitro and in vivo, and even after completion of that wet lab studies, we can validate it using again informatics. So before and after, we can use this informatics. Okay. So when I'm telling this an amalgamating subject. Okay. So it can mix with any other branches of biology also. It can mix with cell biology, it can mix molecular biology, immunology. Take any biology, it can able to use all these concepts. Okay, so protein structure prediction, validation, phylogenetic analysis, drug discovery and virtual screening, network analysis, system biology, immunoinformatics, and peptide-based vaccine design. So three PhD scholars who are working under my mentorship. Okay, two life science students and one technology student. And I also um, recognized in different universities as a supervisor in an university, advisory committee member in Tech, and doctoral committee member in VIT and the University of Madras. Okay, as an examiner, and in Sri Ramachandra Institute of Higher Education Research as an eminent scientist member and research advisory committee. Okay, these are some of the projects, like union projects. Okay, what are the subtitles we can work? There's some information on that, but even if you get this information, one has to practically sit and practice each and every step. So project is like a project. I think you believe that we can't able to complete in 2 days, but the tips I can provide you some kind of a tips. What exactly we can work in the form of a project. Okay, in protein structure prediction, data selection, secondary structure prediction, validation, biological implications, and so more topics inside what exactly we can do after the structure prediction. So we can go for tertiary structure prediction, go for model evaluation. We can after that we can use it for docking. We can go for binding affinity calculation, biological insights. We can predict the binding sites. We can go for publication and presentation. Of course, in each and every project, even if I'm guiding projects to the other engineering colleges or science colleges, I anticipate a publication. So why I'm charging minimal is that we anticipate publication even from outsiders. And even whatever grants we secured from Government of India, I have a publication, at least one publication. Okay. If I get a numerous grant, then I might have a minimum three publications. Okay. So that's most important. Just working on many project won't give you evidence. Okay. If and if you publish in journals or book chapters are just a review, isn't it? So try to publish in repeated essay index journals. Now, if you see this phylogenetic analysis, okay, so if you're working on genetic, genome related information, then one can find this application of using phylogenetic analysis. Okay, so like these are the topics: gene or protein selection, sequence alignment, phylogenetic tree construction, tree visualization, interpretation. This is generally the steps in phylogenetic analysis in basic. But in advance, what exactly we can take? So just using phylogenetic analysis won't give you a kind of a project. So we can find, okay, you can go for maximum likelihood analysis using different tools, compare. Because I showed you that each and every tool is showing a different kind of a result, then you can find a kind of find accurate model, okay, by comparing one software with another software, okay, in a particular method. And molecular clock analysis try to use the molecular clock analysis and co. This is an additional thing. If you want to use, you can use it. That is, instead of constructing key using distance and characters, if you want to additionally use the time, okay, then of course you have to use this additional feature, molecular clock analysis, co-evolutionary analysis, advanced visualization, biological interpretation, publication, presentation, drug discovery and visual screening. Drug discovery and visual screening, we can go for drug database exploration, ligand preparation, protein ligand docking, analysis, visualization. This is general thing. I think we are already taken how to practice with Autodoc. We can also practice Autodoc PYRS. Okay, so either today or tomorrow. And drug discovery and visual screening. Yeah, so one should know about docking, PSAR, pharmacophore modeling, MD simulation. The MD simulation actually it takes more time, but this is most important. Even without in vivo studies, a PhD scholar can able to submit a thesis uh, by inclusions through inclusions of in silico studies. In silico studies should be complete uh, if you complete simulation after the docking. Okay, so in silicon in vitro is sufficient nowadays for analysis. Okay, so one should know about the MD simulations, but it takes a lot of time. Okay, so from for 4 to 5 steps you can complete within few minutes, but if you want to do the last few steps, it takes a few days time to complete. May even it takes up to few weeks also. But the simulation, I suggest you not to try with your laptops. Try only with workstations or servers. Okay, or it will trouble you. Okay, the laptop will go too slow. You may get a horrible experience. So try with the workstations or servers. So I'm not taking in deep about this, but I can give you some kind of a clue how to work. Okay, so free energy calculations, drug candidate evaluation, like predicting ADMET characteristics. Okay, using the Swiss website, we can find so many kind of things like stability, binding affinity, toxicity, everything you can find from the Swiss website. I show you that also shortly. Biological interpretation, publication, presentation. This, of course, a larger area. Many of them are trying this. Even CDRA, Central Drug Research Institute, NIPR, many are focusing on these areas. They're not leaving these concepts. Network analysis and system biology. So we have two popular system biologists in IIT Madras. Okay, so working on precision medicine for types of disease like cancer. Okay, system biology approach. In system biology approach, one has to know about the protein-gene interactions, gene expression data, protein-protein interactions. So that is a major data we take. How to construct such a kind of a network. They start working from that. They dig from the scrap and create the networks. Okay, but when compared to India, okay, the developed countries like USA, okay, the top US university ranking universities, okay, are doing well. Okay, even I found Sweden is best in system and on working on systems biology. Okay, but in India, so whatever networks are created, which are available in database, we are using it using Cytoscape visualizer. We are using it, we are interpreting, we are using various plugins, and we are getting an outputs from various plugins in the Cytoscape. Okay, we are going for network analysis using this network analysis software Cytoscape. Visualization and basic analysis. So this is about project. Okay, and network analysis we can also find in advance, like not only just limiting up to protein-protein network, but also we can try to construct diseasome proteome network. Diseasome network is a different one, like a genome diseasome. That is how you link one disease with another disease. Generally, if you see autoimmune diseases, that is psoriasis, asthma, rheumatoid arthritis, are all autoimmune diseases. Psoriasis, arthritis, is there any kind of a relationship between these two? Psoriasis is something like a skin disease. Arthritis is pain inside, isn't it? In the joints. In the joints, we find more pain that we call it as an arthritis. If so, these diseases phenotypic variation is different. But if you see both the disease, you find if you find the top 10 proteins, na, so nearly 8 to 9 proteins are the common disease proteins in both the diseases, except one or two. Got it? So what you understand, there is some kind of a networks even in autoimmune diseases. Psoriasis is linked with arthritis. Arthritis is linked with asthma. The genes responsible for causing these diseases are associated. Even the cancer is like that. In cancer, epidermal growth factor receptor, that is a common target receptor for almost all the types of cancer, for all the organs. For breast cancer, it is an important target. For liver cancer, it is an important target. For lung cancer, so some kind of a genes, okay, or interlinking a different disease. So one should also know how to construct the diseasomes. So genome era started and almost came to saturation in 2005. In 2003, human genome established. Before the 2001 and 2002, rat and mice genomes are established. So everything established in 2005. And after 2005, the scientists in the world focused on developing proteomes. Now, after it happened around 2005 to 2010 or 12, something like that. So the proteomes also involves in developing the protein-protein interaction networks. So whatever the networks developed got saturated in 2015, and from 2015, 1,000 genome projects, 1 lakh genome projects came into existence. Okay, in 2018 to 2020, they got saturated. Now many scientists are trying to focus on diseasome networks. This is the quantum research happening on in each and every few years. Okay, the trending is changing in bioinformatics. Okay, so even if you take this recent trends, okay, one is developing more kind of a networks now. It's started with gene interaction, gene regulation, gone to the proteome networks. Now we transcriptome networks, interactome networks. Now we are going for diseasome networks. Okay, we also have many. We can also work on various research projects in artificial intelligence and machine learning approaches, AI and ML approaches. Okay, so these are some of the projects like prediction of protein structure using AI techniques. We already have AlphaFold. If you are, we have already an AlphaFold software which already predicts, and even the data are present in PDB website. But if that structure is not known, whatever I'm going to work, either in AlphaFold or in X-ray or NMR, if the protein structure is not yet established, is not present in PDB website, out of 2 lakh 20 structures, then I mean out of 2 lakh 35 lakhs 35,000 structures. 2 lakh 35,000 structures are there. I think 2.32 lakhs are there. So if the structures are not there, one can also use the AI techniques to predict the protein structure. Okay, and identification of potential drug targets using artificial intelligence based virtual screening. That is, if you have a drugs, the drug database, you take like an best example, NCB PubChem, drug bank, etc. If drugs are not there, okay, whatever synthesized the drugs you synthesized in the laboratory, if you don't have any 3D structures, I suggest you to go with modeling that 3D compounds and then use the AI techniques to check the best inactive with your target receptor. AI generally used to screen the inactives. The actives generally already used as an API, active pharmaceutical ingredient. So what we call it as a drug. But if the synthesized compound is not established as a drug, you can use AI techniques. Predictive modeling of drug side effects. using bioinformatics and machine learning. Okay. So just it's finding the drug side effect. That is, instead of targeting a single receptor, if you're focusing on multiple receptors, okay, there's receptors which can give you a kind of a side effects. Okay, this can be studied by modeling and binding studies. So not just a modeling, even you have to go for binding. Okay, so for this you can also use bioinformatics and machine learning. Okay, like taking this can also be done through ligand-based approach alone instead of receptor-based or hybrid based, even through the ligand based, we can able to complete it. This is equivalent to QSAR, quantitative structure activity relationship. In in QSAR, we determine the activity of unknown compound from the training data set. But if you want to find toxicity, instead of QSAR, one can go for QSTR, quantitative structure toxicity relationships. Okay. The activity is calculated as the negative logarithm of MICs in microbiology experiment. If so, the toxicity also we can have a kind of a correlation with the physicochemical properties of the inhibitors. Got it. So one can able to use QSTR for the third approach. Fourth, integration of biological networks for drug repurposing using machine learning. Okay, so drug ligand protein interactions. If you drug protein interactions can also be constructed in the form of networks. We have a database called StitchDB. So if you know how to construct, okay, if you know the graph theory, if you know how to use the softwares, we can able to construct the networks. So how to construct? Of course, the wet lab techniques has to be performed. Okay, so we have to go for wet lab techniques. So whatever wet lab techniques we take, okay, that data we use for construction of drug ligand, the ligand protein networks and comparative genomic analysis. So I told you I'm a DC member of a PhD scholar from Anna University who is drafting a plant genome which is not yet established. So then how she is fairly focusing on to establish a genome working in a private engineering college because that plant genus, okay, with a different species, the genomes are available. But for the particular species of the genus, what she is trying is not yet established. So she is going to go for a comparative genomic analysis for even knowing about the evolution and even to get more genomic connotation. Okay, that can be done. Okay, of course, for only after genome sequencing, we get a genome, but one has to compare with the existing genus species. Then they get a more knowledge on that. That's what I call as an annotation. The genome annotation can be done like that. Okay, and machine learning classification of microarray gene expression data for cancer subtype identification. So machine learning classification, that is microarray gene expression data, we find data in the form of ESTs, express sequence tags. What is express sequence tags? The cDNA, the cDNA expression in the microarrays. Okay, so what is cDNA then? Actually, it is a product. Okay, this product we got from mRNA. From mRNA, if you go for reverse transcription, yeah, can give you the cDNA. So one can find mRNA is the basic sample. If you take the basic mRNA, even if you see the SARS-CoV-2, the mRNA vaccines is what we are using as a vaccine, isn't it? Not only for the viral, for treating viral infections, even for treating cancer, this is most important. Okay, the mRNA we identify which is causing the cancer. Okay, which is specific to a patient. If and if we identify, then we can go for a specific a gene targeted delivery. Okay, for this also you need a kind of a machine learning technique. We have types of machine learning, basic types, the classification, and regression, etc. We generally go for classification approach and analysis of single cell RNA sequencing data. NGS, NGS, okay, using in next generation sequencing, and you can use either supervised or unsupervised methods. I think these are more recent researchers. Okay, if and if you learn in IITs or Indian Institute of Science, okay, they will have the workstations and you can work on these kind of specific topics, say 8, 9, 10, including 7. Now, I take you a new topic called immunoinformatics. What is immunoinformatics? This is also called as computational immunology. It's a field of science that encompasses high throughput genomic and bioinformatics approaches to immunology. You know, immunoinformatics, one should know basically to develop a vaccine instead of a drug. Then, what is the difference between a vaccine and a drug? A vaccine is generally a biological preparation that induces acquired immunity against a particular pathogen. You also know vaccine in general can give a prolonged immunity when compared to a drug. If you want a prolonged immunity, an extended kind of an immunity, one should require an adjuvant in a vaccine. If you have an adjuvant, it can stimulate not only stimulate immune response, you can find antigen more immunogenic. So the antigen which is present in the vaccine is going to provide us immunity. If you want to make antigen more immunogenic, you need adjuvants. The adjuvants can also be a plant-based. I am working on plant-based adjuvants in my lab. Even in my PhD research, I completed, and many students have worked on the plant-based adjuvants. And majorly, I worked and presently working on dendritic cell-based vaccines, dendritic cell-based vaccine, which is a professional antigen presenting cell. The DC we call it as like that. Okay. So, and these vaccine adjuvants can also increase the antibody production, promoting humoral immunity, and it can also promote the cellular immunity by generation of specific cytotoxic T cells and memory T cells. So one should know immunology, okay, to know how these to know how the reverse vaccinology approach work to develop a vaccine for a particular virus, or even you can develop vaccines for cancers, types of cancers. Okay, so you know the types of vaccines in school level. You might have seen the classes, classification of vaccines, and these vaccines are used for treatment of what kind of viruses. You know, in reverse vaccinology, the first vaccine developed is for meningococcal polysaccharide. Okay, that is a component they used to treat bacterial meningitis. If you want to relieve from bacterial meningitis, okay, we can use this meningococcal polysaccharide vaccine. So for preparing the meningococcal polysaccharide vaccine, okay, the initial study they take and they find is the computational studies, that is reverse vaccinology approach. Generally, in vaccinology, what we make, we take a virus or we take a component of a virus, we produce it in bioreactors, we multiply, amplify. Whatever we amplify, we just pack it in vials and we supply. We can also take it from bacterial source or even the parasital source to produce a vaccine. This is generally classical vaccinology that is growing from pathogens. So what we make inactive pathogens, or we can use even attenuated pathogens as a component for the vaccines, or even we can destroy the virus membrane. So whatever components present inside, the subunit proteins can also be used as a vaccine. But what is reverse vaccinology, which is computational approach? Instead of taking the major sources of bacteria or viruses, we can take the genome. From genome, we start developing a vaccine, or from proteome. So these are the constructed ones, isn't it? The genomes established in up to 2005, or proteomes established up to 2012 or 15. This we take and we can try to develop a kind of a vaccine. So for this, we should know the structures of the proteins and the other biological molecules. We work on reverse vaccinology approach, try to get a kind of a vaccine. So this is something reverse. Okay, from the established data, we are bringing a vaccine. Okay, not directly taking from the viral source or bacterial source. So what I'm telling, the methodology, the work plan, if you see, we just start with the genome or proteome sequence, and from that we take the antigen selection from B cell and T cell epitopes. These are the two major epitopes we predict for a vaccine discovery. Okay, one can whatever you predict from the computational techniques. Initially, try with the preclinical studies, that is through small animals, rat mice, or even try with the hamsters, uh, also with the rabbits. Okay, in India, and then we can go with the molecular epidemiology. That is first try with the phase one and phase two. That is with the healthy volunteers, and phase three and phase four with the diseased volunteers. Okay, so from computational, we started and we can end up with a drug or vaccine which can also get FDA approval. Okay. And reverse vaccine, for the SARS-CoV-2 vaccines, in 2020, nearly 89 vaccines came, including Covishield, Covaxin. So many vaccines came, but up to present, no vaccine has got an FDA approval. So each and every vaccine is failing in some trials. If it's failing either in phase three or phase four. So it is not coming up to the post-marketing surveillance. Still, it is a contemporary research that is bringing a vaccine for SARS-CoV-2. Why I'm telling still we don't have an approved vaccine from FDA, just because the SARS-CoV-2 spike glycoprotein is experiencing so many mutations. We have alpha, beta, gamma, delta, so many strains of SARS-CoV-2 are existing at present. 100 strains are there. So whatever vaccine developed, Covishield, Covaxin, is for the first strain. How about for the second strain, third strain, fourth strain? The efficacy gradually reduces. I think in Delta strain, the Covishield and Covaxin efficacy is around 16 percentage, 15 to 16 percentage, not crossing 18 percentage. That is 18 out of 100 patients are getting cured or getting prevention because of vaccine. Okay. So one has to develop a vaccine specifically for the Delta strain. If uh, the patient is identified, diagnosed with the Delta strain of SARS-CoV. So for this, still I can utilize this kind of reverse vaccinology approaches. Okay, so this is a kind of a computational chart. Okay, how to use reverse vaccinology, and this is the first vaccine. Okay, discovered from the reverse vaccinology approach, meningococcus B vaccine. But we are also having vaccines for the malaria, dengue. So many vaccines came in this decade, isn't it? From 2020, so many vaccines came, and of course, the computational techniques are playing the major role uh, for the upcoming of new vaccines. Can be viral source, can be for the viruses, for targeting viruses, or even for combating cancer. Okay, this is the publication actually for the Nipah virus, which is established in the reverse vaccinology approach. I tried discovering both T cell and B cell epitopes, but I found B cell epitope for a target protein. I target protein what I used is W protein. There is a W protein that I targeted here, but the other proteins, for all the other proteins, including B cell epitope, was published in the papers. So this is the only gap I found in the loop. Only one gap. Okay, that is the T cell epitope based vaccine for W protein of Nipah virus was not yet done. So just I tried with it. Okay, and I got multiple grants. Okay, not only got the intramural and TNSST grant, and also this was awarded as the best project. Only two projects came to the finalist for INE award. So this is one of the project. Unfortunately, the students can't able to go to the New Delhi to present it. If they present it, they might have secured a grant. I mean, they might have secured a nation award. It happened at that time. So because of unavoidable circumstances, they can't able to attend. And remember, the mentors can't able to attend. Only the students have to attend. So select the students accordingly. If and if the students try to attend, you can select the students. Okay. Okay, structural proteins. If you see the SARS-CoV-2, these are the structural proteins. Okay, and we have also have non-structural proteins like proteases, polymerases comes under non-structural proteins, which is coming in at 1 to 60. So one should, if you're going for a vaccine manufacture, okay, first vaccine design, okay, first you should understand the genome of a particular virus. Then only you can focus. And even if already the papers are published, don't try to do the same. Don't mimic their research. Try to do some other viruses or try to do the uncovered protein targets. Okay. So that is each and every project should have a novelty. So multi-tope vaccine actually, multi-tope vaccine, this is the successful vaccine. The Covishield for the first strain of SARS-CoV-2, it has given 76 percentage, around 76 percentage efficacy. Covishield is just 76, and Covaxin is 82 percentage. That is, no, no, no. The Covishield is around 76 to 82 percentage, and Covaxin is around 90 to 91 percentage. This is the efficacy we got for first strain of SARS-CoV-2. But this the Covax you didn't hear, actually this vaccine is US based. Only the US people utilize this. So they discovered this vaccine using peptides. This is a multi-tope vaccine. The efficacy, it's more than 99 point something. So almost all the patients get prevention. Okay, so from the SARS-CoV-2 infection, 99 point something. But Covishield and Covaxin is not crossing more than 91 percentage. Covaxin is coming up to 95 maximum in few countries, but Covishield just limiting up to 82 even for the first strain. If you take the second strain alpha, beta, gamma, that's it, ruled out. So one should also realize this importance that when compared to the expensive monoclonal antibody preparation and even the less targeting small molecules, one can go with vaccines, especially multi-tope based vaccines. Okay, the multi-tope based vaccines can target more precisely uh, for the viral infections. Okay, they target more precisely for the viruses. So when instead of focusing on the mono MABs and small molecules, one can try with the vaccines. The best example is COVAX. So one should know about immunology on how the T cell epitope, how the T cell receptor is interacting with antigen presenting cell. So I was telling that my PhD research is on studying the different stages of dendritic cell development. Okay, the dendritic cell is a professional antigen presenting cell. So the dendritic cell can give immunity, but one in thousand cells is a dendritic cell. So if you find one in thousand cells or thousand immune cells as a dendritic cell, it is difficult to give immunity against a virus or cancer. So then which is giving major immunity? So if you see the immune cells, major we have T cells. But even T cells are dormant if they are not interacting with any antigen presenting cells. The T cells have to interact to get both cellular immunity and to get also humoral immunity. So this immunity is most important for targeting the cancer or viruses. Got it? So you can use T cell epitope based vaccine or B cell epitope based vaccine. There are even subclasses in T cell and B cells. So based on that, even the epitopes are classified. So these epitopes are used for gives an idea to go for discovery of multiple epitopes. Whatever multiple epitopes you discover, you can link with together and use it as a vaccine for a specific viral strain or specific cancer patient. Okay, so this is something about reverse vaccinology. One should know about the immunology. Okay, that is the signal transduction, cell-cell interaction. Okay, so this one should know how a T cell is interacting with a B cell, what kind of molecules, protein molecules are taking responsible. Okay, so this is important. And in my PhD research, I have taken 165 protein receptors. Okay, and I also tried with a few plant molecules, and I found which plant molecule is best giving a kind of a immunity. So multiple docking studies are done, and even with the multiple docking studies, I have established a database. SVCE has developed an indigenous database to come for a solution for upcoming cancer or viral diseases. We have already developed a kind of a database, but that is a plant-based adjuvant database. An adjuvant is a component of a vaccine. Okay, if you want to use an adjuvant for a vaccine, okay, I just developed a database for not for a vaccine, but for a plant-based adjuvant, a component of vaccine. For this, I have tested all these principles. I focused on a particular cell, focused on cell interactions. Okay, and then I gone with the plant-based and I checked how about its interactions, etc. So one should know about MHC1 and 2 pathways, which is useful to discover the T cell epitopes. Okay, one should know about the structure of these immune proteins, MHC1 and MHC2, which is majorly present in antigen-presenting cells like dendritic cells. If you know, generally the peptide-based vaccines can be discovered. Okay, so more immunology is needed for that. So there is a pre-existing database. There's a large immunoinformatics database, also immunological database. We call this as IEDB. The expansion is Immune Epitope Database. Okay, in this immune epitope database, you can predict T cell or B cell epitope, and you can discover these epitopes for any types of diseases. So you can discover a vaccine epitope for infectious disease, allergic disease, autoimmune disease, and even for cancers. So these are the four diseases they are mentioned. Okay, we have plenty of filters. So not only you can find an epitopes for the disease, you can also find for a specific protein molecules. And even not only we have a deposited data, already they have tried and deposited the information. You can find it for humans. You can also find it for mice and other non-human primates. Even for dog, camel, if you are a veterinarian, if you want to discover a vaccine for a disease, for an infectious disease, you can try initially with it. We have this much data present in IET. So this most important database for working on immunoinformatics. So database is there, and in the right hand side we have tools also. Using the tools, you can interact with the data present in the database. Okay, IEDB. Okay, this is the contemporary researchers I'm working in my lab. And I think I also informed you earlier that each and every PhD scholar is going to get around 20,000 per month. Okay, that is a stipend we provide. In addition to that, I am already working on funded projects. So the expense of chemicals, glasswares, and reagents, I'm taking care. Okay, also anticipating one more grant. So you are welcome to register in Anna University under my guidance. Okay, so you get a kind of a stipend. Okay, you also get a nice opportunity to work on funded projects. I have worked on IL-2 adjuvant, IL-4 adjuvant, GM-CSF. These are actually immune proteins, interleukin proteins, and growth factor proteins. So I have worked on this. And I also worked on development of AH03 adjuvant, and even dendritic cell itself can work as an adjuvant. Okay, these contemporary researches happened in the past decade. Okay, so even the Nobel Prize winner of 2015, I believe, Ralph Steinman, who is the father of dendritic cells, okay, has awarded Nobel Prize in 2014 or 15. Okay, and even a series of Nobel Prizes, consecutively 2 or 3 Nobel Prizes are based on these kind of research. So one is, I think 2017, one person got for uh, the CTLA4 based approach. And okay, so whatever immune proteins they worked, I worked, okay, even the same concepts they worked, but that is well established by the Nobel Prize scientists. Okay. So I just tried with a few approaches. That's it. Many people are trying all over the world, isn't it? But I experienced like whatever I'm working, I found Nobel Prizes are awarded in the globe. So this is the indigenous database, adjuvant immunoanalytical database. My PG scholars, okay, from biotechnology, in has used C sharp as the programming language, and of course, this is a framework. Actually, of course, we linked with the personal homepage. The personal homepage is like server language. Either we can use PHP or even we can go for ASPX, active server pages. Okay, so we can use these languages. Okay, one has used ASP, the other is has used PHP. So this is a first student project. Actually, we had like whatever talking interactions we got from Discovery Studio, we just added these data in the database through SQL. And if you want to search any particular data, we can search. If you are getting a new data, we can update the same, and we can find the same in the form of a table format. For example, if you want to find which is the best adjuvant for rheumatoid arthritis, I can able to get the information through search. If you want to get for which is the best for liver cancer, I will be getting a plant-based adjuvant for the liver cancer. So such a kind of a data we have deposited, and we got the information. Okay, one should know about even I know just we are mentioning that that even the multitope-based vaccine, which is a product of reverse vaccinology approach, is showing the better efficacy, isn't it? So one can also try for the upcoming infection strains. Okay. So I've completed discussing about immunoinformatics. Okay. So the latest trends, that is, I worked on system biology approaches, system pharmacology approaches. So this and virtual screening using Autodoc Pyrex, PYRX, we'll be discussing it on tomorrow. Okay. So if you want to learn these skills, you can attend this. But anyone have any doubts in the present illustrations on mini project? If you have any doubts, you can ask me. And if you have doubts even in your projects, executed projects, that you can ask me at present. Okay. And but if you want to learn more, okay, sit in my lab. We have a simulation servers. We have a shared workstation from IIT Madras. Myself, I have got a fund and collaborated with IIT Madras, working parallelly. Okay, so I'm working on computational studies in IIT Madras and working in vitro and in vivo studies. We have approved animal house facility in Sri Venkateshwara College of Engineering, Sriperumbudur, which is close to Chennai. Okay, so we can also, when and where do we have to submit the project? There is no need to submit the project. The project, it is intellectual property. The, your project is your intellectual property, like a publication, like a manuscript before getting published. No need to submit any projects to me. But if you have drafted a kind of a project, like a work plan for your project, something like that, if you want me to correct or if you want me to provide suggestions, I can provide suggestions. Okay. A project can't be learned in one or two classes, but whatever you learn from the first class, okay, and if you have an idea, okay, then only you can able to draft a work plan for your project, a timeline and a work plan. Got it? So you're just learning the basic skills of bioinformatics in the previous 20 classes. Okay. Even the project, how the UG student is going to work, they are going to work minimum 4 to 6 months, isn't it? That's it. Okay. So you should have an idea that UG student is going to work on 4 to 6 months, and PG student is up to a year or more than a year. And PhD student for a project is going to work for minimum 3 years to how many years we don't know. It's an infinity years. It's happening in many industries. Even after 18 years, no, the scholar is not at submitting, but still he is putting that still PhD bar. That is still doing PhD after 18 years. I'm working on one project related to network biology, and I have some doubts because it's new for me. If you have a doubts, you can stay back and you can share your screen and you can ask me if it is confidential. Uh, if okay, you can let me know. Okay, so even we can have a private meet also. Okay, so I can provide you initially my LinkedIn profile. You can link with me. So chat with me in the LinkedIn. So just okay, get an appointment with me. Okay, so the message in LinkedIn. Then you can we can sit and work on. Okay, if you have any doubts. And anything else to be discussed, I already mentioned for other colleges I'm guiding, even if you have already registered. So we can not only I can help you in computational, but also in vitro and in vivo. We have facilities. We have good of infrastructure in our institution. Okay, so also this is top 10 engineering colleges in biotechnology. Okay, that's what we got from Times of India. Recently, we have posted that in as an advertisement. So if you want to learn, okay, want to work with a project, want to collaborate with me, you are welcome. The faculties are welcome, and so even students are welcome. Okay. Anything else to be left to be discussed? If nothing, tomorrow we'll be discussing on the Autodoc PYRX and system biology approaches. Okay, like the articles what I published in system biology approaches. Okay, how I use methodology. So in detail we'll be discussing. Okay. So thank you very much for your patience and interaction. Even if you have any accommodated doubts, you can ask me. Tomorrow is the last class. Tomorrow is the last class. We are winding off this entire course on tomorrow. And based on your request, your coordinator, Professor Praveen Ramanujam sir, is going to schedule an MCQ. Okay. So you can appear for MCQ based on your request. I think he has already informed you. MCQ is just for your self-assessment. The marks are not going to be present in the certificate, or you are not going to get any grades in this certificate course, but still try your best. Try to learn, okay, before getting appearing for the test, for the MCQ. Yes, tomorrow is the last. When will the MCQ test occur, sir? Only on tomorrow you can come to know, sir. Tomorrow or day after tomorrow. Okay. So I think he gives sufficient time based on the feedback received from the participants, most of the participants. He fixed a date for you to conduct MCQ. Okay. So don't worry on that. Okay. So it's based on your feedback only. When you request, the date he fixed. Okay. Anything else to be discussed? If nothing, okay. Thank you. Have a great day to you all. Good night.